Welcome. The first question very commonly asked in a job interview for a production officer role in the pharmaceutical industry is a request for the candidate to introduce themselves or provide some information about their background and qualifications. Question is introduce yourself or tell me something about you. Always answer this question in simple, short, and specific words. You can change yellow highlighted part and make your answer ready. My name is Praveen Kumar. I am from Mumbai. I have completed my B Pharm from Mumbai University and I have 3 years of working experience in pharmaceutical industry. Second very commonly asked question is tell me about your industrial experience. I have worked in ABC Pharmaceutical Company from Jan 2021 to October 2023 as an production officer. I have handled various equipments required for batch processing like autoclave, while washing machine, while filling machine, etc. You can add your specific experience details and make your answer ready. What is your job role in current organization? I am currently working as while filling officer in the ABC Pharmaceutical Company. My job role involves operating and maintaining the vial filling machine to ensure the accuracy and efficient filling of vials as a part of the production process. Here also you can add your specific details and make your answer ready. What is the role of production officer in pharmaceutical industry? Production officer in the pharmaceutical industry is responsible for performing, supervising and managing the production process, ensuring compliance with regulatory standards, maintaining quality and optimizing production efficiency. Which are the important guidelines that are referred in pharmaceutical industry? Some important guidelines or regulations include good manufacturing practices, that is GMP, Food and Drug Administration, that is FDA's guidelines, and International Conference on Harmonization, also known as ICH guidelines. What are good documentation practices or GDP in the pharmaceutical industry? Good documentation practices or GDP refer to the set of guidelines and procedures that ensure accurate, complete, and consistent documentation throughout the pharmaceutical industry. What are the good manufacturing practices or GMP in the pharmaceutical industry? Good manufacturing practices, that is GMP, in the pharmaceutical industry are a set of guidelines and regulations that ensure the quality, safety, and consistency of pharmaceutical products throughout the manufacturing process. What is a change control? In simple words, change control is the documented, established procedure to control changes in the pharmaceutical industry. FDA defines it as change control is a systematic approach to managing all changes made to a pharmaceutical product or system. ICH-Q10 defines it as a systematic approach to processing, evaluating, approving, implementing, and reviewing the changes. What are the types of change control? As per ICH Q10, change controls can be classified into following 5 important classes. First, minor change, second, major change, and other 3 are temporary change, emergency change, like for like change. What is deviation? In simple words, departure from established procedure is called deviation. Established procedures are referred to standard operating procedures or standard test procedures, batch manufacturing records or batch packaging records, approved protocols like qualification protocols, validation protocols, etc., and any other approved document. Failures to follow instructions given in these established documents leads to deviation. What are the categories or classifications of deviation? In pharmaceutical industry, deviations are classified into following 3 categories. First, minor deviation, second, major deviation, and third, critical deviation. How do you classify deviations? There should be rationalized approach for the deviation classification. When deviation does not affect any quality attribute or critical process parameter, it would be categorized as minor deviation. When deviation causing probable impact on product parameter that will impact on patient health shall be classified as major deviation. When deviation is having highly probable impact on patient health and cause danger to patient's life shall be categorized as critical deviation. What is difference between corrective action and preventive action? As per ISO 9000, preventive action is taken to prevent occurrence, whereas corrective action is taken to prevent recurrence. Consider the case of machine breakdown of machine A. If we identify and implement actions that will avoid recurrence of machine breakdown on machine A, then it will be falling under the category of corrective actions. If we identify and implement the actions that will avoid similar occurrence of machine breakdown on all available machines, example machine B, machine C or machine D, that actions will fall under the category of preventive actions. What steps do you take to prevent cross contamination in pharmaceutical production? Here you can describe your procedures like cleaning, equipment segregation and following control strategies as per SOP. What is ALCOA++? As per WHO, MHRA and FDA's document, ALCOA stands for A. Attributable, L. Legible, C. Contemporaneous, O. Original, A. Accurate and ++ signs are for complete, consistent, enduring and available document. When we should perform equipment qualification? Equipment qualification shall be performed in 3 cases. First, at the initial introduction of equipment, premises or system to the facility. Secondly, as per the periodic requalification frequency. And third, when major change is done to the qualified equipment, premises or system. What is area qualification in pharmaceutical industry? Area qualification is the process of evaluating and documenting the suitability of a specific area within a pharmaceutical facility to ensure it meets regulatory and operational requirements. Commonly referred guidelines for area qualification are FDS 21 CFR Part 211 and the EU GMP Annex 1. What is calibration and why is it essential in the pharmaceutical industry? Calibration is the process of verifying and adjusting the instrument accuracy. It is essential in pharmaceuticals to ensure accurate measurements, maintain data integrity and compliance with regulatory requirements. One more question related to calibration. What are the main parameters that require calibration in the pharmaceutical industry? The main parameters include temperature, pressure, flow, pH, conductivity, weight and time. What is the limit for airborne particles concentration, that is NVPC, in grade A, grade B, grade C and grade D? You can refer below table for limits for airborne particle concentration for the monitoring of NVPC, that is non-viable particle contamination. This table is from UGMP Annex 1, point number 9.15. This one is about clean rooms. Which guidelines are commonly referred for clean room in pharmaceutical industry? ISO 14644, UGMP Annex 1, US FDA's guideline, PICS guideline, WHO guideline ICH Q7A ASTM E2500 and ISO 14698 are commonly referred for clean room in pharmaceutical industry. One more important concept in pharmaceutical industry is cleaning validation. So basic question is, what is cleaning validation? As per PDS technical report number 49, cleaning validation demonstrates or procedure to ensure that the cleaning process adequately and consistently removes product residues, process residues and environmental contaminants from the cleaned equipment or system. One more question related to cleaning validation concept. Question is, when we should perform cleaning validation? Commonly in pharma industry, we perform cleaning validation or verification in 3 frequencies. First, cleaning validation should be conducted for the initial qualification of a manufacturing process or equipment. Second, further periodically cleaning verification shall be done. And third, in case of major change in the cleaning procedures or equipment trend, we should perform or consider cleaning validation. This one is very important question for the production officer in the interview. Question is, why 3 cycles are considered during validation or qualification run? Answer is, consecutive 3 cycle data gives enough statistical information to conclude on the repeatability of results. 1 or 2 batches data may found insufficient, and obtaining data from more batches probably will give repetitive results. Here additionally some guidelines like PDS technical report number 29 also suggest to take 3 consecutive cycle runs during validation. Cleaning and disinfection both are the very important concepts in pharmaceutical industry. So question about them is, what is difference between cleaning and disinfection? Cleaning is done to maintain a visually clean and tidy environment and removing non-microbiological contaminants, whereas disinfection is done to reduce the microbial load on surfaces and prevent the transmission of infectious agents. One more question about the differentiation. Question is, what is difference between sanitization and sterilization? First we will see sanitization. Sanitization refers to the process of reducing the microbial load on a surface or equipment to a safe level. It does not necessarily eliminate all microorganisms, but it reduces them to a level that is considered acceptable for the intended use. Whereas in case of sterilization, sterilization is the complete eradication or inactivation of all viable microorganisms, including bacteria, viruses, fungi, and spores. The goal of sterilization is to achieve a total absence of viable organisms. One more important question, what is the difference between sterile and aseptic area? First we will see about the sterile area. Sterile area is an area that is completely free of microorganisms and contamination. It ensures the highest level of cleanliness and safety for sensitive processes like sterile drug manufacturing. Whereas aseptic area is an area where strict procedures are followed to prevent contamination and maintain cleanliness. It is important to note that aseptic area may not be entirely free of microorganisms like sterile area. Question about the HEPA filters. Question is, what does HEPA stands for and what is its primary function? HEPA stands for high efficiency particulate air. Its primary function is to filter out particles and microorganisms from the air to maintain clean environment. What is the efficiency of a HEPA filter? HEPA filters typically have an efficiency of 99.97% for particles as small as 0.3 micrometers. You can see below guideline reference for this information. Why 0.2 micron filter is used for aseptic filtration in the pharmaceutical industry? A 0.2 micron filter is used for aseptic filtration in pharma industry because it effectively removes a wide range of microorganisms aligned with regulatory requirements and has a history of successful use in maintaining product sterility. Reference PDS technical report number 41 2008. Question about electronic signature. Electronic signature is a set of symbols that is as unique and legally binding as a handwritten signature, but that is used to sign records in a computer system. Question about human errors evaluation and control in pharmaceutical industry. Why human error evaluation and control is important in pharma industry? The reason behind its importance is the great majority of reported defective medicinal products has resulted from human error or carelessness, not from failures in technology. Reference MHRA Orange Guide 2002. What are the 5 common reasons for human errors in pharma industry? 5 common reasons are: first, lack of training and education. Second, communication breakdown or communication barrier or communication error. Third, fatigue or overwork. Fourth, complex processes and systems. And fifth, distractions and multitasking. Next question, what is HVAC qualification in the pharmaceutical industry? HVAC qualification refers to the process of verifying and documenting that heating, ventilation, and air conditioning systems in a pharmaceutical industry or facility are designed, installed, and operated according to predefined standards and regulatory requirements. What are the critical environmental parameters monitored during batch manufacturing process? Critical environmental parameters include temperature, humidity, pressure differentials, and particulate count. Reference for this information is UGMP Annex 1. Which QMS elements trigger investigation? Following are the 6 important QMS elements against which we perform investigation in pharma industry. First, deviation. Second, OS or OOT results. Third, market complaints. Fourth, audit observations. Fifth, self-inspections. And sixth, non-conformance reports. What are the key elements of investigation report? Key elements of investigation report are problem in brief, immediate actions, investigation team details, review of previous history, investigation details, root cause, CAPA actions, impact assessment, summary and conclusion, attachments and post-approval. What is pH? Full form of pH is potential of hydrogen. Lower pH value indicates a higher concentration of hydrogen ion and higher pH value indicates solution with lower concentration of hydrogen ion. At lower pH value solution is more acidic and at higher pH value solution is more basic or alkaline. Which are the simple investigation tools commonly used in pharmaceutical industry? Simple investigation tools includes brainstorming or group discussion, report from SME that is subject matter experts, YY analysis or 5Y analysis, personal interrogation, review of previous history of non-conformances and process mapping tool for investigation. Which are the advanced investigation tools commonly used in pharma industry? Advanced investigation tools includes 5M or 6M tool for investigation, fishbone tool for investigation which is also known as cause and effect diagram or Ishikawa diagram. Next one is failure mode effect analysis, and last very rarely used tools are Pareto charts and scatter diagrams. What are the reasons for lower assay in pharmaceutical industry? Here are the 7 reasons which are commonly associated with lower assay in pharma industry. First, incorrect dispensing of API. Second, incorrect handling of API. Third, error during volume makeup. Fourth, product degradation. Fifth, inadequate mixing. Sixth, sampling error. And seventh, analytical error. Which are the 5 reasons for using stainless steel grade SS316 as product contact part in pharma industry? Here are the 5 reasons for using SS316. First, corrosion resistance. Second, biocompatibility. Third, sterility maintenance. Fourth, temperature stability. And fifth, regulatory compliance. Which are the 5 reasons for using epoxy in pharmaceutical industry? Here are the 5 reasons for using epoxy in pharma industry. First, chemical resistance. Second, hygiene surface. Third, ease of cleaning. Fourth, durable coating. And fifth, regulatory compliance. What are nitrosamine impurities and why are they a concern in the pharmaceutical industry? Nitrosamine impurities are potentially carcinogenic substances that can be found in pharmaceutical products. They are a concern because of their potential risk to patient health. What is the significance of conducting stability studies for pharmaceutical products? Stability studies assess the product's shelf life and how it degrades over time, ensuring the product remains safe and effective throughout its intended storage period. What is the role of standard operating procedures, that is SOPs, in pharmaceutical industry? SOPs provide step-by-step instructions for performing tasks, ensuring consistency and compliance with quality standards. How is data integrity ensured in the pharmaceutical industry? Data integrity is maintained through the use of secure electronic systems, appropriate access controls, and proper record keeping practices. What is the role of internal audits in pharmaceutical industry? Internal audits verify compliance with GMP and other quality standards, identify areas for improvement, and ensure ongoing quality system effectiveness. What is the role of training in pharmaceutical industry? Training ensures that all employees are knowledgeable about GMP, SOPs, and quality related procedures, reducing the risk of errors and non-compliance. What is concept of RFT in pharmaceutical industry? Full form of RFT is right first time, doing the things right in first attempt and every attempt. RFT focus on if something is done right first time, it is done perfectly every time and no money and time is wasted for correcting errors. What is technology transfer in pharmaceutical industry? Technology transfer is the process of transferring product, process, packaging and analytical knowledge from one site to another site. Product technology transfer is transfer of product and process from transferring site to obtaining FDA's or regulatory approval. What is temperature mapping in the pharmaceutical industry? Temperature mapping involves monitoring and recording temperature variations within the storage areas or processing areas such as warehouses, cold rooms, autoclaves, incubators, product processing areas to ensure product quality and compliance with regulations. What is US FDA's expectation about training in pharmaceutical industry? US FDA expects that each person engaged in the manufacturing, processing, packing or holding of drug product shall have education, training and experience or any combination thereof to enable that person to perform the assigned functions. The US FDA has identified GMP training requirements in pharmaceutical industry and defined in 21 CFR 21125. What are the weighing balance calibration parameters? As per USP chapter number 1251, following are the weighing balance calibration parameters. First, sensitivity. Second, eccentricity. Third, linearity. And fourth, repeatability. Last and very useful question, what are the important regulatory authorities for pharmaceutical industry? You can refer this list of 10 important regulatory authorities along with their country name. Best luck friends, keep watching Farm Grow and subscribe to channel for more videos related to our pharmaceutical industry.